since the beginning of the century, when Francisco Mojica discovered CRISPR within the adaptive immune system of bacteria, viruses have been painted as some sort of evil, defeated villain. Your Honor, I'm here today to contest that rumor and sue Mr. Streptococcus pyogenes on behalf of my client, Mr. Bacteriophage, for defamation. Did you really think that viruses weren't going to have a comeback to CRISPR? Of course not. And this is what we call anti-CRISPR. Before CRISPR-Cas9 can cut the correct piece of viral DNA which it finds with its guide RNA, anti-CRISPR will bind to Cas9, effectively preventing it from cutting. But let's take a look at how exactly anti-CRISPR completes this task. Here are the 6 domains of CRISPR-Cas9. The PAM interacting domain binds to and recognizes the PAM, while H&H and RUBC each make a cut in the double-stranded DNA. There are 2 types of anti-CRISPR proteins that effectively inhibit SpyCas9, ACRIIA4 and ACRIIA2. ACRIIA4 has shown greater rates of inhibition and promise. We will be mainly focusing on that in this demonstration. Anti-CRISPR deactivates Cas9 by causing 4 important interruptions to its mechanism. First, anti-CRISPR will inhibit recognition of the PAM in the PAM interacting domain, which the Cas9 protein will not cut without due to the risk of cutting the wrong DNA. Anti-CRISPR can do so with negatively charged residues imitating phosphate groups of the PAM nucleotides. A lack of recognition of PAM will also interrupt Cas9 mechanisms and prevent it from unwinding the DNA and then stabilizing the DNA with guide RNA and a phosphate loop. Third, anti-CRISPR blocks the tunnel for non-target DNA, which the RUBC domain is responsible for cutting. Anti-CRISPR physically blocks the active site of this domain, so the protein does not have access to the DNA. This will then cause H&H to stay in its inactive conformation. H&H makes a slight conformational change when Cas9 has bound to its target DNA, so that its catalytic residue histidine can be closer to the phosphate group on the target DNA. Thus, inhibition of target DNA binding would cause H&H to remain in its inactive state. But wait, couldn't Cas9 just choose to bind to the DNA instead of the anti-CRISPR? Well, anti-CRISPR competes for the spot on Cas9 with DNA and has actually developed and evolved a higher binding affinity towards Cas9. But there's no need to fear. Just because anti-CRISPR inhibits Cas9 well doesn't mean us humans are all doomed. In fact, a rewarding partnership of use between CRISPR and anti-CRISPR may be in the cards for us. One of the main reasons why scientists cannot employ widespread CRISPR-Cas9 treatments yet is because of its lack of accuracy. And this lack of accuracy is understandable. Faced with the enormity of the human genome, Cas9 can sometimes cut off target. Recently, scientists are looking to anti-CRISPR to selectively inhibit Cas9 when it cuts off target. Furthermore, many are looking to anti-CRISPR to help during Cas9 treatments targeted at a certain area or tissue. Anti-CRISPR can be injected into neighboring areas to prevent Cas9 from spilling over and cutting DNA that we don't want it to. Scientists have already found extraordinary promise in using this technique. In closing, Your Honor, due to all the benefits that humans stand to gain from the hard-earned evolution of viruses, Mr. Streptococcus pyogenes should be found liable for all counts of defamation against my client, Mr. Bacteriophage. Thank you.